Hello, science enthusiasts. My name is Jason Zakowski. I'm a high school chemistry teacher and science communicator, but I'm also the dog dad of Bunsen and Beaker, the science dogs on social media. If you love science and you love pets, you've come to the right place. So put on your safety glasses, do up your lab coat, hold on to your tail. It's time for the Science Pawdcast. Hello, everybody. Welcome back to the Science Pawdcast. It's exciting to be back. It's the first episode of Season 4. We have an entire year of amazing folks who will talk science to you. Plus, every single week, we've got the breakdown of science news and the science that's going on with our pets. Plus, we'll give you the lowdown about what's happening with Bunsen and Beaker. We have some really exciting news. The Telespark Center has partnered with us for a Bunsen and Beaker night. There's going to be a meet and greet for folks to come meet Bunsen and Beaker, which still is really hard to wrap my head around. And I'll be doing the Science Pawdcast live. Another interesting development, which is kind of cool, is our spaces on Twitter have really taken off. Twitter has this audio chat room function. It's called social audio. They're called Spaces. And we have a program we run on Twitter called Spaces Unleashed. And it's become quite popular. We were just named to a top 22 Spaces hosts to follow list, which was really humbling. There's some amazing Spaces hosts out there. Our patrons get access to the recordings first, but we've been rolling them out to our podcast feed. If you subscribe to the podcast, you'll be seeing Spaces Unleashed bonus content, and those are our live chats from Spaces. One show is about science, and the other is about pets in general, the dogs, and people share their pet stories. It's been a lot of fun and super rewarding to connect with people via voice on Twitter. On the show this week, in science news, we're going to talk about the James Webb Space Telescope. It got to the L2 point, but I thought it would be great to talk about what it is, what it's going to do, some cool tech that it has, and what the heck the L2 point is. In pet science, we're going to talk about some of the reasons why it's important to train your dogs just for fun. This week in Ask an Expert, we have Dr. Yane Valdez, immunologist, to talk to us about the immune system and a whole bunch of other fun topics. She's an absolute riot. I can't wait for you to listen to the interview. Hey dogs, what do you call a telescope that can't stop running into things? A kaleidoscope. Okay. You know we had to start season 4 with a great pun. Okay, on with the show. Because there's no time like science time. This week in science news, let's chat about the James Webb Space Telescope. I get a little antsy and excited about all things space, and it was pretty nerve-wracking to see it launch because it had been delayed, and I've been teasing Dr. Jessie Christensen, who is a NASA scientist who's been on the podcast before, because it seemed like every time she tweeted it was going to launch, it was delayed. But it did launch. I think it launched on Christmas while we were on break with the podcast. And nerve-wracking, it got to space great, and it's been traveling to this L2 point, Lagrange 2 point, and it basically reached it a couple days ago. So that would be, well, what day is it today? January 29th? January 28th or 27th, I believe, it reached the L2 point finally. And NASA had a cool little tracking app where you could watch it as it moved towards the L2 point and the different kind of like maneuvers it had to go through, and the scary kind of like unfolding of the mirrors. And my students were watching along with everybody in the world. Every day they came into class, they'd ask to see where it was, and we check, and a little bit of anxiety to see if it was still there. But I thought we should talk a little bit about the James Webb Space Telescope. Not everybody knows a lot about it. Like, what is it? Why is it different? What is it good for? What is the L2 point? All right. When are we going to start getting pictures from it? So this is just a mini breakdown about what's going on with the Webb telescope. Well, the first thing I should probably mention, and I was a little bit, jump the gun a bit. I thought we would be seeing pictures within a couple months, but it might be around 6 months before we get the first couple pictures. It's reached it's kind of like its place in the solar system to take pictures, but it's got to go through a whole bunch of different, very complicated startup procedures, double checks, because if anything goes wrong with it, it's in a position that we're not going to be able to go fix. It's going to be way too expensive to go fix any issue, so they have to get it right the first time. So the second point is, what the heck is the L2 point? Now, it's pretty technical if I get into the math, and I did read a lot about it, but I'm just going to parse it down with probably a couple main points. The L2 point is the second Earth-Sun Lagrange point. Now, it's about 1.5 million kilometers from Earth towards Mars. So that's how far the Webb telescope has gone from Earth, 1.5 million kilometers. It's basically where the sun and the Earth's gravity balance out, where the inward pulling force of both the sun and the Earth are relatively the same, which keeps a very small object on a curved path. Why is this important? It means that whatever's put in the Lagrange points, and there's 5 of them, the L2 point is where the Webb telescope's headed for, requires very little fuel to keep it in that circular path. It can sit there, and the tugging forces of the large gravitational bodies are somewhat equal. Now, it still needs fuel. This is something that I didn't know at all. I thought once they stuck it out there, it was kind of like it was like a little hole it fell into and it could just sit there. No, it needs fuel to maintain its orbit around the sun, and it's eventually going to run out of fuel, which is shocking. Like, I did not know this, but it has a lifespan of about 10 years. NASA, of course, always undershoots the lifespan. So I think they're saying it's 10 years. But my guess it's going to last a lot longer than that. I know they have to say, if everything is used as inefficiently as possible, it's got a 10-year lifespan. It's on borrowed time, this amazing space telescope. It has to use fuel periodically to maintain its orbit. The main reason why it's so far away is that some of the technology on it has to operate at extremely cold temperatures. So the Webb has to be far enough away from things that give off energy, like the Earth. The Earth actually radiates energy into space, so that would affect Hubble, and it would affect the infrared spectrum. So the telescope doesn't want to give off more energy than the energy of light that it's absorbing, so it doesn't kind of like mess up the wavelengths. So that's one of the reasons why it's got to be really, really far away. One side of the telescope has a shield to block sunlight and reflect it back. And the other side of the telescope is this super, super cold surface, minus 233 degrees Celsius. So one side, one of the engineers said would be so hot you'd boil water on it from reflecting all the sunlight. And the other side, you're very close to absolute zero. Now, some of the instrumentation on it, one's called MIRI, that's the mid-infrared instrument. It uses further coolant to bring it down even colder. It takes the coldness of space and makes it colder because space isn't cold enough, I guess, to make this instrumentation work. All of the mirrors on the one side, you've probably seen them. They're gorgeous. These hexagonal segments, I think there's 18 of them. They are going to focus all of the light that it records onto a detector plate. Those 18 segments will act as one 6.5 meter long mirror. That's a huge mirror. They have to, of course, align all of those individual mirrors perfectly to whatever they're going to look at. So it's a whole procedure to look at stuff in outer space. Now, some of the technology that really sets it apart has fun acronyms. One is called the NIRCam. And the NIRCam is the infrared camera that will cover the ranges of wavelengths of light in micrometers. Like we're talking like very accurate cameras. And this is going to be able to image the earliest stars and galaxies right after the Big Bang. 12 billion years ago. So it's going to be able to, this technology will give us a snapshot back in time of the earliest things that current technology can observe, way earlier than Hubble or anything else that we have on Earth. The other thing that's super interesting and I'm really excited about is that this NIRCam can look at stars and measure the dimness as exoplanets go around it even better. It's called the coronagraph. It also can look at objects in our Kuiper belt. One other cool piece of technology it has is called NIRSpec, and that's a spectrograph which will look at the same light waves as the NIRCam, but instead of like imaging it, it's going to turn it into data about what makes up the image's properties, such as its temperature, its mass, and its composition. and it can look at 100 things at the same time. This is mind-boggling. MIRI is a mid-infrared instrument, and that's the one that's got to be kept super, super cold. MIRI has a camera and a spectrograph, and this is going to be used to also look at distant galaxies and new stars, but it could also spot planets, comets, and asteroids. The last technology I think I'm going to mention is something called the FGS. So the FGS and its partner is called the NIRIS, Near Infrared Imager and Sightless Spectrograph. Again, that is specifically for exoplanets. So there's some technology to image things that are really far away. There's technology to figure out what the stuff really far away is made of. And then there's specific technology to image exoplanets. Whatever they look at first is being kept under cloak and dagger. On Twitter, I was actually in this huge NASA room where they were talking about the Webb telescope, and I got to ask NASA a question, probably because of the size of Bunsen and Beaker's account, and that we do science communication. And I asked, who decides what they're going to look at? Like, did they play darts or pool? Like, did they go bowling? And it was kind of funny. But the answer is it's a secret, and people submitted papers anonymously to determine which object the Webb telescope would look at first. That way they didn't know if it was a grad student or a PhD candidate or some NASA scientist. It was all anonymous. They've selected the first object, and it's going to be a secret and a surprise. I can't wait for it to come out, and I hope we all learned a little bit more about the James Webb Space Telescope. That's science news for this week. This week in Pet Science, we have some great advice from Dr. Zazie Todd, who's an animal behaviorist, and she was a former guest on the Science Pawdcast. This comes from her blog, and I'll have a link to it in the show notes. And it's all about why training dogs is so important, broken down into 8 easy-to-remember reasons. The first is it's fun. Obviously, if something isn't fun, it's a lot tougher to do. I can tell you from experience, as many people can, it's a lot of fun when you train your dog. It can be frustrating when they're not getting it, but when they get it, it's a lot of fun. And by and large, most dogs, they just want to figure out what you want them to do. They really want to do it right. They want to figure it out, and the gears in their head are turning. Bunsen was always trying to please us, and Beaker's brain's like a computer. She's trying to figure out what we want. So it is a lot of fun. The other reason is it builds your relationship with your dog. It's true. Spending time with your animal outside of them lounging on the couch or you taking them for a walk, where you're actually working with them through positive reinforcement, is more likely to cause a strong attachment with your animal. The third reason, it's good for your dog's welfare, which means that if you're using positive reinforcement, it's a great way to give them that good experience in life with humans. All right, this is so true. Another reason why it's important to train your dog is it tires them out. It absolutely does. After training, Beaker's beakering is at 0. She has to sleep. She's just cooked. She's so done. Making the dog's mind work is important. Most dogs were bred to solve tasks, and doing that training is a task they have to solve. Another amazing reason why you should train your dog is it helps with cognitive enrichment. It gives their brain something to do. This is the reason why you have puzzle boxes for their food, or you give them Kongs. It gives them a little bit of extra stimulation in their day beyond chilling on the couch or the floor. It's great for their brain. The sixth reason it's important to train your dog is it gives them food enrichment. This is where your dog might get a little bit of extra nutrients, extra fats from the special treats that they get. If you've trained your dog, you have low, medium, and high value treats. And you generally, for simple tasks, try to get the dog to only get, you eventually train them so that the low value treats is what they get for simple tasks, and tougher tasks or new tasks that they figure out, they get the high value treats. And those high value treats can be things with really high protein. Beaker loves fish. So we have these little freeze-dried fish, and she only gets those when she's training. So it's a little bit of food enrichment. Another interesting note is that the reason why you should train your dog is that your dog gets better at learning. The more you train your dog consistently, the faster they get at learning new tasks. And our dog trainer, Shar, said that with Beaker. She's like, you need to keep the momentum up when you're training your dog. They understand the routines. They figure out the tasks faster. They don't lose the old tricks and tips. So it's great to keep momentum, and your dogs will get better at training the more that you train them. Lastly, and most importantly, and this is true for Chris and I, it's important to train your dog because you, the human, practice proper skills in dog training. I don't know how many times with Beaker in the last little bit, me personally, or Chris, we have been the ones that have failed her at trying to learn a new trick or trying to learn a new obedience thing. We haven't positioned our body right. Our hand signals were ambiguous. Our voice was the wrong tone. And learning that from somebody who uses positive reinforcement and has a lot of experience trains you in how the dog learns best. And that's great for you to learn. Like, Bunsen's been through all that, but it's amazing how much you forget as a person and how to train a dog. Like the hand signals need to be crisp. You need to clearly articulate to the dog what you're doing. So that's another great reason why training your dog is important. So while we don't have any science articles this week, Dr. Zazie Todd's points about why you should train your dog are all backed up by science. I thought it was a fun way to start season 4. That's Pet Science for this week. Hey everybody, before we get to the interview section, I thought I would just give you some ideas about how you could support the Science Pawdcast. Number 1, you could support us on Patreon. Check out patreon.com/BunsonBurner. There's multiple tiers of support, and the lowest tier of support is not much more than a cup of coffee a month. The second way is you could check out our merch shop. We've worked really hard to partner with clothing companies that do a great job of providing vibrant colors and soft feels. We also have the Beaker Stuffy for sale. It's so cute. The third way you could support us is giving us great reviews on our podcast playing apps. Any kind of review helps. And if you can't find a review, share our podcast with people. Thanks, everybody. Now back to the interviews. It's time for Ask an Expert on the Science Pawdcast, and I am delighted to have Dr. Yane Valdez, immunologist, with me today. How are you doing today, Yane?
I'm doing well. Thank you. Thank you so much, Jason.
Where are you calling into the podcast from? Where are you in the world?
I am actually in Vancouver, British Columbia. This is the traditional, ancestral, and unceded territory of the Squamish and Musqueam nations.
So, how long have you been in Vancouver? Are you there for work, for speaking, for your job, or have you lived there for most of your life or your whole life?
Actually, I'm originally from Peru.
Right?
I came here 25 years ago and thinking I'm going to study and go back to my country. But I ended up here. I'm here for the past 25 years.
So you lived in Vancouver for 25 years, or Canada, or...
Yeah, that's correct.
Oh, okay. All right. Do you go, I know COVID's tough to go anywhere. Have you been back and forth to your where you were born?
No, I didn't go back to Peru. I was planning to go this year actually to celebrate the 200 years of independence of Peru, but COVID didn't allow us to move, so we're here still. Yeah.
You have a doctorate in, and you're an immunologist. Could you talk a little bit about what your training is in science?
Sure, definitely. So I have a master's degree in biochemistry and molecular biology, and I also have a doctorate in microbiology and immunology. So I got into science because of my curiosity, I guess, and my love of learning. So yes, I studied in Cusco in Peru, in my hometown. I studied biology because I was fascinated by evolution. I thought I was going to be a paleontologist, and eventually
work with dinosaurs.
Work with dinosaurs. I think I'm fascinated about evolution in general. I remember in high school, I was intrigued by metamorphosis and the transition from tadpole and adult frog. So I was like, this is what I want to understand. What happens, how the genes. I mean, yeah, early enough, very early enough in my life, I learned that I wanted to be a biologist, but I didn't know that I was going to end up as an immunologist.
What was the thing when you were young that hooked you into science? Was it like a science class? Was it looking at pictures of dinosaurs?
Actually not. It was, I think, my dad's teaching. My dad had enormous patience for teaching us, and he will always love nature. So he will be teaching us experiments and about insects, about plants. But I remember very well, he had a magazine, I think it was a Time magazine, something like that, and discuss about evolution. I do remember clearly, I think I was probably 8 or 9, there was a picture of the DNA there. And my dad used to say, oh, here is where the genetic information is. I didn't understand that point, what genetic information. But every time that I will talk about evolution, I will always relate to that molecule of DNA that I had pictured in my brain. So, yeah, it was more about the family, even though I don't have any family that now, look, in the past had I would say, a science background. I think from my dad, after that, my brothers, for instance, my brother is a trauma surgeon, my sister is an ecologist, and myself an immunologist. So I think it was a lot my dad's teaching.
Sometimes it's an adult in our life that shapes us and what we want to become later. So that's a touching memory and story. Then you started to work with Helicobacter pylori. Did I say that right?
Helicobacter pylori.
Helicobacter by pylori. Could you talk to us about what you worked with that? Like what is that and what you were doing with that stuff?
I think I was drawn to working in infectious diseases after witnessing the devastating effects of the cholera epidemic in my country. So I felt a call to make a difference. And somehow I found myself studying this bacteria, Helicobacter pylori, that has high incidence of infection and reinfection and recurrence in my country. So for the context, H. pylori is a bacteria that induces gastritis, ulcers, and is associated to gastric cancer. So in my group, I was leading a small group in Peru. So we wanted to understand why the regular therapy of antibiotics was not effective in Peru, because there were many cases of reinfection. So my work was at the level of, I would say, molecular epidemiology. So what I would do is take biopsies from different parts of the stomach from different patients. So one patient, different parts of the stomach, get a biopsy, grow the bacteria, then isolate the genetic material, in this case the DNA, and then compare the DNA profiles of the samples. So we discovered that.
Cool.
So we discovered that actually people have a mix of H. pylori strains in the same stomach. So in different parts, there were different kinds of genetic material. And
what? It's like they have different environments or something? Or like
yeah.
Oh, cool.
So, the stomach has different, like we'll say the ones that are associated to the esophagus, the ones that are associated to the intestines, and they have it's the shape itself, But the problem was that we found that we had mix of antibiotic susceptibility strains in different parts of the stomach. So then no matter what the treatment was, it was not useful. So even despite using a mix of antibiotics, so there was always a prevalence of antibiotic resistant strains. So that for us, teach us that we need to have a different thinking in how to treat patients, So we proved that the conventional therapy that works in the developed country didn't work in Peru.
Oh, so like one antibiotic just didn't do the job. And if that was the main treatment, it was not working
No,
great because it
no,
would leave some of them left alive because they're
exactly.
different.
Different. So
Oh.
then we tried different therapies, and yeah, so that also makes you think about what we call like personalized therapy. Each
Mhm.
person it's different, and the disease one person might not reflect the disease of the other person because we are different individuals. So yeah, so that was interesting. That was in H. pylori in Peru. When I came here to Canada, I actually didn't work in H. pylori. I work in Salmonella, which is
Oh, can you talk to us about that? I love because salmonella is one of the foodborne illnesses that
yes.
everybody blames for everything. So sorry I jumped in there. I just can't wait to hear your
You're
take
fascinated
from
about Salmonella, I
yeah,
guess, with me.
yeah.
Yes. Yes. So I did my PhD here in Vancouver in the laboratory of Dr. Brett Finley. So this is a laboratory in which you can have freedom to pursue any area of infectious diseases. So I chose to work in Salmonella. As you said, it's this bacteria that induces food poisoning. So I was very, very curious to know what happens early in the intestine. So when Salmonella enters the gut, what happens at that moment? So eventually, I really use animal models, and so I understand what type of cells of the immune system. We have the pathogens, but at the same time, we have an immune system that defends us from these pathogens. So I wanted to understand which are the first cells and how do they fight this pathogen, this infection. So I studied one cool cell. It's called dendritic cell. That cell lives underneath the epithelial layer of the gut mucosa. And this cell is called sentinel because it's there to grasp. It has dendrites like hands, like fingers, and grab the bacteria and test it and said, this is a pathogen, this is not a pathogen. It's a cool cell.
What it grabs it? It grabs it and it's like, hey, what are you? Like
yes, it grabs it.
what?
Between the epithelial cells, sends a hand, grabs the bacteria, and
No
eats it.
way.
Yes,
That is wild.
It's wild. It's cool. It's really cool. So once the sentinel cell, these dendritic cells, recognizes that it's a pathogen, then starts sending signals to other cells. So and calls other cells. I think. So I learned that one of the first responders cells are called neutrophils. These are like really kind of like funny cells. They run immediately when they're called. And in my studies, it was so cool because I did some staining to see those cells, and actually they make walls are like grabbing each other so they don't let the bacteria go inside the gut. So they restrict the bacteria in the gut and not allowing to get into the other organs, because if the bacteria gains access to inside to other organs, you have a really more difficult disease. So then this allows, and then your body start flushing out. That's why you have diarrhea and this.
Oh yeah.
So that is actually very, very cool. And seeing which cell comes, how they come, how they defend is fascinating. Yes.
It's so cool.
Yeah, it's so cool. It is so cool. But Salmonella is also very sneaky. Salmonella. Salmonella like guts. I was like, okay, now they found me this way. I'm gonna cover myself this way. I'm gonna send this way. Things like that. So it covers, for instance, what we call the endotoxin. It covers, mimics, changes, tries to hide. It's interesting. So, but also for us was really cool to understand what are the weapons that Salmonella has. So because Salmonella sometimes lives inside the cells and hijacks the cells. And so we want to know what are those weapons. And in the lab, we all worked. And we actually developed a vaccine against chicken Salmonella. So the idea that we get if we vaccinate the chickens, so the chickens won't get salmonella, so the eggs and the chickens won't pass to humans. So so it's fascinating. It's I love it. I love understanding this interaction between the pathogen and the immune system. One brings this, the other brings that. It's just fascinating.
I could listen to you talk about the immune system all day long. Like I'm making like little mental pictures of these cells with grabby hands. They're like, what are you? And then you're not supposed to be here. And the other guys just come booking it over, and they're like, traffic jam, you don't get in. Like it's like they've got little personalities and specific jobs. I love it.
They do. Yeah.
So cool. With salmonella, just one quick follow-up question. People get very, very sick with salmonella, and it can lead to death, can it? Like it can kill you if you're not careful.
Okay, correct. And there are at least two major types of diseases. One is called gastroenteritis, and that is a self-limiting disease. So you suffer for a couple of days, but as I said, those cells, they don't let the bacteria go inside. So then the bacteria is still in the lumen of the gut mucosa, so then it's eliminated. However, if you are immunocompromised or you have another bacteria called Salmonella typhi, that Salmonella actually could go inside your organs. It could go into the liver, into the spleen, and it's fatal. So then for that, you need antibiotics because bacteria shouldn't be inside. Like mostly it's it's outside, it's in the gut, in the skin, but inside we don't have much bacteria, so the body reacts and mounts a huge immune response. So then that could be lethal. But the like self-limiting food poisoning could be lethal in people that are immunocompromised, in kids, in young kids. But for adults like us, will make us miserable, but perhaps not that, causing death. But yes, it could, but it's not as the typhoid disease.
Yeah, it does make us miserable. I've had food poisoning a couple times in my life. I don't know if it was salmonella, but it was over as fast as it started. So I get your self-limiting thing. I'm glad it was that way with me.
Yeah.
So Janet, from your work with salmonella, you have now moved on, or in your career, you moved on to a time with something called stem cell technologies. Could you talk to us about that?
Sure. So, Stem Cell Technologies is a biotech company, and so I joined Stem Cell Technologies in 2014. At that point, Stem Cell was the largest biotech company in Canada, I would say.
Oh, wow.
And conveniently located in Vancouver. So,
that's nice.
Yes. So I joined the cell separation team, and I led the innate immunology team. So what was my job? My job was to develop these tools to help scientists isolate specific blood cells, so from blood, from tissues. What we did is just use magnetic particles that we target to different antibodies, and then we can use a magnet to separate the cells. So then my job, yes. So, for instance, you have the blood sample. So then I will get a particle. I will link it to an antibody against, we'll say, this neutrophil that I was talking. So then I will put that sample into a magnet, and I will capture all the neutrophils in this.
Okay.
And then wash it, and then you ended up with your neutrophils. So then you can work with your neutrophils. So. Yeah, so that was my job. So then I led a group of, 9 to 10 people. And so that was it. I was making these tools and help scientists. So that was it.
This is a hard follow-up question. I don't know if you can answer it. Is from your time there, is there something people could wrap their heads around? Like, what does that lead to? How does that help, kind of thing? Like, I'm sorry if that's a stupid question or a bad one, but I'm just curious. I mean, it's cool. You're using magnets to like separate out the neutrophils into, that's cool. Go ahead.
Yeah, no, it's in the lab when we're working, for instance, if we want to ask specific questions about a cell type, so we put the cells into different environments, we trigger them with different molecules, so we want to understand the behavior. For instance, if you want to say, okay, these dendritic cells, how this dendritic cell reacts to this virus. So then you, in vitro, what you do is you get the cells, and then you can start putting a virus and then see the responses. So for that, So in vivo, obviously, the important thing is in vivo, but it's sometimes very difficult because there are many, many signals happening at the same time. So if you want to dissect the specific signals or the specific way one cell type behaves, so then you do that in vitro. And for that, we were developing those so we can distinguish the response from, we'll say, the dendritic cell, the neutrophil, the macrophages, and then start asking specific questions in vitro. Then you can start moving to animal models and then see how they work together. Yeah.
So in a general sense, and I might be a bit off base, but just to kind of bring it to something topical, the new COVID variant, Omicron, Pfizer just released a thing saying that in their lab tests, the antibodies seem to do okay at the third dose level in vitro, not in vivo.
Vitro. That's correct.
Vitro. Okay, yes. So in the lab, but you'd have to like do something like the magic you're doing to get the antibodies away from all the other factors, right? So it's a very specific question.
Correct. So
did I do okay, or is that an
you did okay? Oh, you did
A for effort, F for science? I don't know.
I'm understanding.
Okay.
So yes, that's what I did. So I learned so much though. I learned how a biotech company works from making business cases. This is a business case. What are the specific roles of departments like manufacturing, quality control, marketing, sales, etc.
Yeah.
From developing the product in the lab all the way to put it on the shelf. It's a huge process.
Wow.
However, my heart is more at the level of discovery rather than development. So although it was a rapid and important learning experience for me, I returned to academia. So I feel that I'm more fulfilled, and my values, my passion for science, I think I can contribute I can make a biggest difference there in academia and less in industry. So that's why I'm back.
Gotcha. But with that experience, you now have like an extra thing in your Swiss Army knife of abilities. Like without that experience, you wouldn't maybe be the same person you are right now.
Absolutely. I think it's so important to actually see, because you imagine things, but it's not the same as going through the whole process. When people say, oh, develop a vaccine. Yes, you can develop a vaccine. But now what happens if this vaccine will have to go to billions of people? So that's the other part in which biotech is so important. So for me, that I was there, even to do a small like cell separation products or media, it took months. How it has to be absolutely robust and clean product. So that part people don't understand until you go through the whole experience. So for me, it's one of the things that I do is learn from every aspect, from academia, industry, and now I can see with different eyes, different perspective. Yeah, definitely.
Wow, I'm just blown away. You're just so inspiring and so passionate. So I'm so glad I'm talking to you, and you agreed to be a guest. Like, this is just a fantastic conversation.
Thank you.
I can just hear it in your voice, and it's getting me inspired too to learn more. So now that you're away from that company, you're kind of into a different area of your career, which sounds just as fascinating. Let me start that again. You travel around as an invited lecturer. Like that sounds amazing. Can you talk a little bit about that?
Yeah. So I felt when I came to Canada, many people helped me, and I always wanted to contribute to the society, because that's what, as a society, we work. So I had this in my mind all the time. So then I was very fortunate that in 2019, I joined the faculty of the Immunology Without Borders, which is a chapter of the International Union of Immunology Societies. So my husband and I actually joined. So we went to Cotonou in Benin. So the faculty is composed of scientists from around the world, and our students were from Africa, mostly West Africa, but all around from Africa. So we taught different aspects of immunology, and also teaching the students how to write grants and present them. So we were there for a week or 2 weeks, from 8 to 8 working. So this was a rich experience for me. I always wanted to go, as I said, wanted to give back to society. So I was very fortunate to give back to students in developing countries, and because all these classes are free. So we volunteer our time, and they're for free. So I think, education should be free. I feel that I contributed in that sense. So because this was so incredibly rewarding for me, so I started teaching immunology in Latin America. So I did it by Zoom, and because of the pandemic.
Right, yeah.
And hopefully I will have the opportunity to go and teach in person. So
I would sit in one of your classes. I would fly there to listen to you talk live. I would hop on a plane today.
Student. Fantastic. I love that. Yeah. So it was great to teach in Latin America. Even though I'm Peruvian, I speak Spanish, but I never really took immunology in Spanish. So I was teaching in Spanglish. So half in Spanish, half English, but we had fun. It was amazing. So,
I would have to brush up my Spanish to sit in your class. I only know the words that Dora the Explorer has said while my niece has been over, so I'm not great there.
No, I think I'm better. You know that immunology has its own language because it's full of acronyms. So that's a thing. Yeah. Okay. So I speak immunology. So I will teach in immunology. Yeah. It's my third language.
And is this something that you're going to continue? This is something you want to continue into the future with? Like, is this the next stage of your career, or you just want to see how it goes?
Actually, I love doing this. But in the meantime, during the pandemic, so we applied to go to Barcelona in 2020 with my family. My husband is also academic. So both of us, we thought we were going to be in Spain for a year. It's the first time our sabbatical. We did everything to go there, apply for schools for my son, everything, get the housing, everything sorted out. But the pandemic came and ruined the plans. So instead, I took my own sabbatical here, and I joined the COVID-19 Resources Canada, which is you probably heard, it's a grassroots initiative that provides the resources to fight the pandemic. So this is in Canada. So what we do is serve as a, reliable source of information and expertise for COVID-19 research. We support and facilitate coordination of research efforts regarding COVID-19, capacity building, public health, research, fight misinformation or disinformation.
Yeah.
So last year, what I was doing is a vaccine question and answer, which is it's a kind of a kitchen table style vaccine question. So we open up for the public. They can come and they ask any questions they wanna here have. And so then we would just reply their questions. So it was a series of experts that will join and a series of people. So then it was free, and people had so many questions about the pandemic. So we just sit and they will ask us the question. So in addition of these vaccine question sessions, we just launched a new initiative. And so I'm leading this program. This program is called Vaccine Conversations. So, So what we're doing is teaching people communication skills to help them have these difficult conversations. Now vaccine becomes such a has this stigma. People don't know how to talk. You don't know how to talk, how to address friends, family, your community. I hear so many cases of relationships, for instance, being put apart by the pandemic, and particularly by the vaccines. So what we're teaching is first have to fight this infodemic. Infodemic is this disinformation and misinformation about vaccines. So we did is we built a massive library of scientist cure repository of verified information. So then it's a group of volunteers. We joined and we actually looked all this, and we put this library. So then it's accessible for everybody that wants to see. So we have materials from infographics to small videos, etc. And what I'm doing now is hiring these facilitators that are people that are going to teach you how to have conversations with people about vaccines, particularly now that the holidays are coming. Many people want to get together. People don't know how to address, are you vaccinated? You're not vaccinated. And so how to have these meaningful and difficult conversations.
Yeah,
so that's
tough.
What we're doing. It is so difficult. Oh my God, it's so.
I need this resource. I think we all do.
You are more than welcome to join our, I think it's evenings, and you are in the mountain time. So so it's for you will be from we'll say 6 p.m. to 8 p.m. It's free. You just go ask your questions. Actually, we have role-playing parts, and this is super fun. So one person plays to be the vaccine, I don't know, denier.
Hesitant?
Hesitant. I don't want to say hesitant. It's just because now it's having this negative connotation. I would just say, I don't know, confidence. They don't have confidence in the new vaccines, I guess. And the other person is the person that advises what to do. So it's super fun. I have a blast every time that I go there in the evening. So more than welcome.
Okay, Janet, just a quick follow-up question. This interview won't come out for a while, probably not till I'm banking them for our next season of the podcast. Will this still be going on next February, March, or is this going to be over really quick? What we're talking about right now?
I don't think so. We are actually just initiating the vaccine conversation.
Okay, perfect. I can cut this out. I just wanted to double check that this wasn't going to be over in December, and then when it comes out, people are going to be looking and be puzzled. That's all.
Yeah, no, I don't think so, because now boosters are coming, and then kids vaccinations. Yes. Mm-hmm. Yeah. And Omicron's here. So no, we have for a while.
Perfect. Is there a link for this that people can find or information that we could put in the show notes that people could look more into it for themselves?
Absolutely. So I can pass the information to you. COVID-19 Resources Canada Vaccine Conversations. That's it.
Perfect. Okay. Gotcha. We'll make sure that's in our show notes for people to have easy access to.
Yeah, perfect. Thank you.
But when the spread of Delta was really bad, our account tried to gently educate people. And it is a difficult conversation. People are very, very upset to not necessarily have what their viewpoint is challenged, but they don't like hearing about vaccine. And some of everybody has family members probably that it's very difficult to talk to them about any of this stuff. just because it's really, really drove a wedge between people. So having more resources like this is incredible. So just on a personal level, thank you.
Oh, no problem. Thank you. Yeah, no, we heard this from many people, and actually ourselves are in the same situation. Everybody that I know have somebody that is not vaccinated, and they don't know how to talk. From really, really family members, close family members, to actually friends or neighbors or somebody in the community. So no, it's something important that we can do. I don't want to see more relationships like killed by COVID. It's on top of having people sick and dead, you have relationships. So it's going larger.
You betcha.
No, I'm not. Anything that I can do to help, I'm there. Yeah.
We're going to switch to the next couple questions, and they're the standard ones on the podcast. The first one is a pet story. We ask our guests to share a pet story from their life. Do you have a pet story you could share with us, Doctor?
Oh my goodness, this is so difficult because I had so many pets. So my home in Cusco, in Peru, was kind of like the SPCA here. So we rescue all the animals. So particularly the ones that their mother died during delivery. So we rescue goats, lambs, and feeding them in milk in bottles. So these pets grew with us, with myself and my 6 other siblings. So it's so hard to choose one, but I guess I will choose my goat.
Aww.
Bertoldo, he was my baby again. I fed him bottles, and he was so mischievous and so smart. Oh my God. Yeah, it was a nightmare, but at the same time, really cool and funny. But I think right now, I guess, I don't know if you saw the picture of my baby. It's called Pisco. Pisco is my dog, our current dog, and it's named after the Peruvian drink, Cusco. So we call him the baby in the house. And he actually believes he's the baby, not only the baby, but the king of the house. So he knows and manipulates us all because we adore him. But he's so gentle and tender.
Oh my goodness, he's so cute. Pisco, there's a picture of Pisco with Toby.
I know. Toby is a friend's doggy that comes and have play dates with Pisco. So Pisco is super, super sweet dog. So and so Pisco is teaching Toby how to be nice and how to play. I think one of the best things from Pisco is that he actually rescued a baby hummingbird. And we named this Hummingbird Elliot. So we were in our house in Saturna Island, and I heard him making noises in his wall, and I went, and it was Pisco licking after this baby hummingbird. So we rescued the hummingbird. We named him Elliot, and So he didn't eat it, nothing. He just licked at the hummingbird. He was so cute. So we knew that he is very, very sweet dog, and he is. And Pisco, he greets everybody in the neighborhood. Human, animals, everybody, super sweet. So anytime Pisco and Toby that comes here sometimes, they're cute. So these are our babies, and you will see in our Twitter handles anywhere, we always put pictures of Pisco and Toby now.
You betcha. We'll make sure also there's a picture of Pisco in the ad for your episode. Yeah, because Pisco's got kind of a Bernese Mountain Dog facial features too, which is adorable.
I know. Yeah,
he kind of looks like Bunsen.
Yes. That's right. Yeah.
That is adorable. Thank you for sharing your pet story.
No.
The other question we ask our guests about is something called the super fact. It's something that you know that when you share it with people, kind of blows their mind away a bit. Do you have a super fact you could share with us?
Okay, so my super fact was, do you know that we are more bacteria than human cells? So your body has 10 times more bacteria than your own human cells. So you are more bacteria than human.
So what is that common with lots of other animals, or is that just a human thing?
It's common with many, many animals. Just again, back to evolution. So when we started to become more and more evolved, we were dragging with us many of the existing life, like we'll say bacteria. Also, your genome might have around 10% viral genome because we were dragging with us. We're like evolving with all these microorganisms. So yes, it's not only human, but also animals and even plants,
Wow. So I guess if we wanted to lose weight, we would just have to kill all those bacteria, but I think we'd get sick.
Yes, you don't want to kill that bacteria. You don't, because some of them, for instance, like we say, vitamin K, C, you don't process them. They do it for you, so you don't want to kill them. In fact, many diseases now are associated to our microbiome or lack of one species or another. So yeah, as we evolve, we took advantage of other microorganisms. Yeah, so they do a lot of work for us.
Right. So they're not living rent free. Is that what you're saying?
No. So we give them food, and we actually use their byproducts. What they use to, we'd say, they eat, they assimilate. If you eat fiber, for instance, and they will multiply, and the products of that, digestion, will enter into our body to help us. There are new studies now in some molecules called short-term fatty acids, which are so important. So pretty much bacteria teaches you how the immune system works too. I mean, they educate you. So as soon as you're born, you are covered in bacteria, and that bacteria is going to help you actually in the future. Like, it's so interesting. As soon as
so cool.
As soon as you come out from your mom, you're covered in one bacteria that we call lactobacillus, which digests milk. So as soon as you start drinking milk, you're covered in your intestine with this bacteria that will help you to assimilate the milk from your mom. So it's fascinating how the evolution. See, that's very important. That's so important, evolution.
That is so important.
Yes. So they not only that, they eventually in the next 2 or 3 years, you're going to be, because you were in a sterile environment at the beginning, then you start getting more and more bacteria that will help you in the future. So
that's right. And toddlers are super grubby, so they're getting all of the bacteria all the time.
That's correct. Yes. So yeah, so you're more bacteria than human.
So we have millions of little friends, even though we don't see them.
Yes. So that's why we have to be really good with them and eat good food for them.
So they can help. Yeah. So they're not living rent free.
No.
And yeah, they're in a symbiotic relationship with us, and we help them out. They help us.
Perfect. That's it.
Perfect.
You put it in a really easy way to understand. Yeah.
Great. That is a super fact. I love it. It might disturb people a bit, but you know what? Some super facts are super interesting and super disturbing to the certain people. So there we go.
Yeah.
The last section of the podcast is a fun one. We get to know a little bit more about our guests outside of what they do. Some guests talk about hobbies, some talk about causes. Janet, you wanted to talk about EDI. Could you speak to that for us or with us?
Yeah, definitely. So EDI stands for Equity, Diversity, and Inclusion. So EDI I guess, mattered to me long before I knew what it stand for. So I am an immigrant, a mom of 2 kids, and I actually saw firsthand how some scientific and social spaces can be unwelcoming to women and people of color. So I do a lot of work in EDI now. So I'm an EDI advocate. I dedicate my time to eliminate the social, cultural, and institutional barriers to women's success in STEM. So I feel that as a woman, we can't afford to lose any more talented, intelligent, curious, and hardworking women. And that's what's happening in now, particularly in the areas of STEM, because we are losing women. Now, even during the pandemic, it's even worse. So we need to address this issue. We need to retain women in workspaces, particularly in the sciences and in the academia. So that's why I'm working. I'm giving talks to bring awareness, not only here in Canada, but also in Latin America and in other parts of the world. So if you, I work with different and help different institutions like the Swiss, the Society for Women in Science in Canada, and different groups of women. So that's where my advocacy is now. So if you have any questions, if you want more awareness, I'm here to help.
And that's why I love having voices like you on our show. And my wife is a big proponent of that too, is like we like to have diverse voices on the podcast. And thank you for talking about that with our viewers. It's something that has been on our radar because it's story after story. Like the people that were hit the hardest during the pandemic were moms, single moms, who didn't have the same kind of support that males had, or single male who didn't have kids on top of whatever else was going on with the pandemic. So it's important to bring light to all that.
Yeah. I mean, and we need different allies. So in your case would be, as you said, you are a white person. So you could be an ally. You could always be defending groups of women and like people of color, those are the ones that like have massively hurt during the pandemic. So I think just to elevate their voices, just to recognize that we are in this together, and it's good allyship. So definitely. So everybody has a role, because so I'm doing this job because of advocacy, but it is better if we get help from many, many groups, because indeed we didn't create the problem. So it's sometimes in our shoulders, but we need to bring awareness. So being an ally it's very, very important. So join the cause. Absolutely.
You betcha. Tell me where to sign up. Is there an organization or a link that you'd like us to share, or just having this message is important for you, Janet?
I think having the message, because everybody could do something in their own networks. I think. Okay. Yeah.
We're kind of at the end of the interview. I wish I could talk to you for hours. You're just a fascinating and inspiring person. I was laughing so hard. I had to mute my mic, and then I started to cough, and then I had to keep my mic muted. I just want to learn more about the immune system. It's fascinating. And also your pet story plus your advocacy. It's just such a great conversation. Thank you for talking to us today, Janet. Where can people follow you on social media? Do you have an account that folks can link up with you? Yeah, go ahead.
Yeah. So I'm only in Twitter. Yeah. So my handle is at Yane Valdez T. So yeah, you can find me in Twitter. I don't have other accounts. I think that's good enough for me. Thank you so much for giving us this platform to share our voices, to know each other. It's fabulous job that you're doing. Thank you, Jason.
Hey everybody, before we get to the family section, I thought I'd just tell you a little bit about this fun thing that we're doing on Twitter. Twitter has this new function called Spaces. It's an audio chat room. You log into the chat, it's as easy as clicking, and you can listen away, and on mobile, you can speak. We run 2 audio chat rooms a week. We're calling them Spaces Unleashed. On Tuesday at 7 p.m., we have an amazing guest that we interview and then open the floor to questions. Some of the guests are from the podcast in previous episodes. On Saturday, we have Spaces Unleashed, Pet Chat, and you can come and listen to the wacky adventures of Bunsen and Beaker, but we really like hearing your own stories about your own pets. If you're not on Twitter, it's a reason to sign up. Come for the Spaces Tuesday and Saturday. In the show notes, you'll find our weekly newsletter that you can sign up for, which keeps you up to date on what's happening in Spaces on Twitter.
Okay, it's time for story time with me, Adam. If you don't know what story time is when we talk about stories that have happened within the past 1 or 2 weeks. I will start. This morning, as I was getting ready to go in the shower, I had a towel on, and Dad came downstairs. I'm like, okay, Dad's downstairs. I got to time this properly so then I don't have an awkward encounter. So I waited for him to go in like the laundry room, and then I went towards my bathroom, and then I hear thump, 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 thump. And I look at the stairs, and it's Beaker, and she's so happy to see me. She has like the biggest smile ever on her face. I have no clue why, but she was so happy to see me. So happy. She was so smiley. And I mean, I play with her downstairs sometimes. Like I run around with her because like she gets traction on the rug, but so happy. And I pet her and she was very huggy. She was very close and she was very cute. But that's my story. Beaker being cute and close downstairs. Mom, do you have a story?
I sure do. But before I tell my story, I want to piggyback off of your story. You may recall that we have been training Beaker in level 2, but the difficulty of that and having 2 dogs is Bunsen likes to train as well. And I needed some quiet time with Beaker to be able to do training just with her. So I took Bunsen downstairs and he got to hang out in your room. And maybe Beaker was a little bit jealous of that. I'm not sure. But my story relates to the kindness challenge that I am doing for 28 days for the month of February. I created a kindness challenge using resources that I found, and I compiled a calendar of things that we can do every day. And I've shared it with my school, and I've shared it with my Twitter followers. But every day, Beaker, Bunsen, and I are doing a kindness challenge because we're going to sprinkle kindness like confetti and make it go everywhere. And that's my story.
Dad, do you have a story?
Adam is hauling Beaker around because she's little and you can pick her up. So, all right, what's my story? It was nice, and then it's super cold again, and that's made Chris very sad. But one thing that's very funny is when it gets cold, somehow Bunsen gets superpowers. He gets energized. He's full of P&V. He is faster than Beaker. He wants to like roughhouse with her in the snow. He jumps forward and smooshes his face through the snow like a weird, some kind of mole, like a snow mole. And he wants to chase you, and he acts like a little puppy when it gets cold. And when it gets colder, he acts even more weird. It's like he starts beakering. So that's my story of Bunsen and his superpowers in the cold. He gets super hyped up and super silly. And that's just, it's really cute about Bunsen.
All right. So that's the end of story time. I'm glad to be back on the podcast. It's been a little bit since we've done that, but yeah, it's a pleasure to be back, and I can't wait to see you guys next time. Bye-bye.
That's the end of another podcast episode. Thanks for coming back week after week to listen to us. Special thanks to Dr. Yane Valdez, who talked to us about immunology in such a fun and entertaining way. Also, special thanks to our top tier patrons on Patreon. Without their support, we wouldn't be able to do what we do. Every week, we read their names at the end of the podcast. If you want to hear your name read, head to the Patreon page in the show notes and sign up. Take it away, Chris.
Chris Kelly, Samantha Dodd, Kimberly Bond, Nate Stephenson, Debbie Anderson, Courtney Proven, Renee Hardy, Mary Rater, Shelby Leggett, Mary Coos, Katya Lynch, Marianne McNally, Andrea Persons, Elizabeth Bourgeois, Karen Beth St. George, Bianca Hyde, Sandy Breimer, Tracy Halberg, Jenny Jaguar, Leela Perriello, Lynn Armstrong, Lisa Swartz, Catherine Jordan, Donna Craig, Lila Ashir, Jody Ogren, Liz Button, Kathy Zerker, and Ben Rathert.
For science, empathy, and cuteness.